In this video, we will discuss necrosis, what it is, how it happens, and its key features. Welcome to MedExplain. Let's get started. Before diving into necrosis, we first need to understand cell death and why it happens. Cell death is an irreversible loss of cell function due to either a programmed process called apoptosis or a pathological injury, that is necrosis. Now let's get into it. Necrosis is a pathological process that is the consequence of severe injury. This injury could be anything like loss of oxygen supply, which is ischemia, or exposure to microbial toxins, burns, or chemical or physical injury. Whatever be the cause, the final outcome would either be inflammation or damage to the surrounding tissues. Now let us understand how it happens. As we know that the cells are projected to injury, this injury causes damage to the membrane of the cell. The plasma membrane eventually breaks down due to the lack of oxygen or any other trauma or toxins. This damaged membrane causes enzymes from inside the cell to leak out that eventually break down the cell itself. This broken cell releases its inner materials like ATP produced by the destruction of mitochondria and uric acid, which is produced by the destruction of DNA, into the surrounding area. These leaked substances act as danger signals, that is commonly called as DAMPs, which means damage associated molecular patterns that alert the immune system. We know that special immune cells like macrophages detect these signals, which ultimately results in the phagocytosis of the necrotic debris. These macrophages, which are clearing up the dead cells by eating them, release certain signals called as cytokines. The cytokines act as inflammatory mediators and cause eventually more inflammation that helps to produce more amount of macrophages and that clean the mess. Together, the digestion by macrophages and immune response produced by the dead cells and cytokines released by macrophages clear the dead cells from the body. This is how necrosis appears in gross. It is swollen due to the fluid built up. It can appear pale or dark depending on the type of necrosis. It can be pale due to lack of blood supply or dark or black due to dead tissues and hemorrhage. It can be soft, firm. Some types of necrosis make tissue very soft and mushy, like a liquefactive necrosis seen in brain, while others keep it very firm, like coagulative necrosis. But eventually, there will be a loss of normal structure. This tissue no longer resembles to that of the normal healthy version anymore. It could also be foul smelling in certain cases if there is involvement of bacteria. This is mainly due to putrefaction. Here you can see examples of coagulative necrosis in the kidney, the right image. Notice how the affected area appears pale and firm compared to the surrounding healthy tissue. This happens because of severe ischemia, that is lack of blood supply, leading to protein denaturation while the overall tissue structure remains intact. Unlike liquefactive necrosis, which is seen in brain, the left image, where tissue gets completely broken down, coagulative necrosis preserves the shape of the organ for some time before the body starts clearing it away. During necrosis, the cell is severely damaged and ribosomal RNA gets degraded. Generally, this ribosomal RNA, it stains blue, that is basophilic in H&E staining, because hematoxylin binds to the acidic structures like nucleic acid. Now, since there is less RNA, the usual blue staining also fades away. The necrotic process causes protein denaturation and eosin binds strongly to these altered proteins. This increases eosinophilia, making the cytoplasm look pink. Normally, the glycogen particles in the cell give it a granular appearance. But since in necrosis there is a loss of glycogen particles, it causes the cells to turn glassy and giving them a homogeneous appearance. Normally, the cytoplasm has a dense, uniform texture because it is packed with organelles. When necrosis happens, enzymes digest these organelles like mitochondria, endoplasmic reticulum, and lysosomes unevenly. As these organelles disintegrate, they leave behind empty spaces called as vacuoles in the cytoplasm. So under the microscope, these vacuoles make the cytoplasm look patchy and full of gaps, similar to how a piece of cloth looks when moths have chewed through it. Now, since the cell membranes are made up of phospholipids and there is a gradual destruction and breakdown of cell membrane and membranes of other organelles like mitochondria and endoplasmic reticulum, there are whole phospholipid precipitates that are formed. They can either be phagocytosed by other cells or can be degraded into fatty acids, which eventually bind to calcium, resulting in calcification. Now let's have a look at the nuclear changes involved. The first one is karyolysis, also called as nuclear fading. The chromatin in the DNA dissolves by enzymes, which results in the loss of DNA. The second one is pyknosis, that is nuclear shrinkage. The chromatin present in the DNA condenses into a very dark and a dense mass. This causes increased basophilia, giving it a blue tint. And the third one is karyorrhexis, where there is fragmentation of the nucleus. Eventually, the nucleus breaks down into pieces and disappears completely. Here is a question for you. Listen carefully. Which of the following is the earliest change seen in necrosis at the microscopic level? Option A, karyorrhexis. Option B, pyknosis. Option C, karyolysis. And option D, increased eosinophilia. Alright, if you know the answer, drop it down in the comment section below. And that's a wrap on necrosis. If this video made necrosis less painful to learn than the process itself, drop a like, it keeps the grind going. Here, subscribe for more no-nonsense medical breakdowns. Oh, and don't be selfish. Share this with your friends who might need a quick revision. See you in the next one. Stay sharp, stay savage.